Kaya mo yan. Chill lang.
Kaya nga nabubulol talaga ako. Ang bilis mo magsalita.
Bilis mo magsalita. Hindi. Sakto lang. Okay na? Nagre-record na ba?
Oo, nagre-record na.
Sige. Good afternoon, everyone. We are here to present... Wait lang, narinig yun?
Hindi.
Akala ko narinig. May dumaan eh. Wait lang, ulit. Good afternoon, everyone. We are here to present the process and the results of the experiment in nucleic acid.
First, what is nucleic acid? Nucleic acids are the main information-carrying molecules of the cell, and by directing the process of protein synthesis, they determine the inherited characteristic of every living thing. Also, nucleic acids are very large and complex organic compounds composed of nucleotides. Each nucleotide is made up of three components: nitrogenous base, pentose sugar, and phosphate. Nucleic acid have two kinds: deoxyribonucleic acid, DNA, which... Ulit talaga! Ulit. Doon lang sa part ko. Sa part ko lang ng first. Pa-umpisa lang. Okay, huwag ka na mag-ano yung intro. Bubulol talaga ako, putik. Yung sa may unang ayan. First, what is nucleic acid? Nucleic acids are the main information-carrying molecules of the cell, and by directing the process of protein synthesis, they determine the inherited characteristic of every living thing. Also, nucleic acids are very large and complex organic compounds composed of nucleotides. Each nucleotide is made up of three components: nitrogenous base, pentose sugar, and phosphate. Nucleic acid have two kinds: deoxyribonucleic acid, the DNA, whose sugar is deoxyribose, and ribonucleic acid, the RNA is with ribose sugar. DNA is the master blueprint of life and constituents the genetic material in all free living things, organisms, and most viruses. RNA is the genetic material of certain viruses, but it also found in living cells, where it plays an important role in certain processes, such as the making of proteins. A qualitative analysis of nucleic acids, based on the presence of phosphorus as phosphate, pentose sugar, and the nitrogen, which are derivatives of purine and pyrimidine. Next is the isolation of RNA from yeast. Step 1 in isolation of RNA from yeast is place 125 ml of water in beaker. Second, add 25 ml of 1% sodium hydroxide and 20 grams of dried yeast. Third, heat the mixture in a water bath for 30 minutes, constantly stirring it while heating. Fourth, remove the mixture from the water bath and filter it with a cheesecloth. Fifth, cool the filtrate and add 3 drops of acetic acid. 6. Evaporate the filtrate up to 50 ml. 7. Cool and pour with vigorous stirring into 100 ml of 95% ethanol with 1 ml of concentrated hydrogen chloride to precipitate the RNA. And the last one is, when RNA has settled decant, dry the RNA in a petri dish at room temperature. The next one is solubility test. What is solubility test? The tests are performed to determine the ability of compounds to dissolve in a solvent, which is usually a liquid. These tests are essential to determine the size and polarity of unknown compounds and the presence of acidic and base functional groups. The solubility test in the example here is cold water, which is insoluble; hot water, which is slightly soluble; alcohol is insoluble; and the dilute hydrogen chloride is insoluble; and the last one is dilute sodium hydroxide, which is insoluble. For the next slide, Ms. Irene Mundo will discuss about the hydrolysis of RNA in acid.
There are numerous chemical tests that can be used to determine the presence of the nitrogenous base, pentose sugar, and phosphate components in a nucleic acid. But before conducting chemical tests, it is very necessary that the RNA should undergo hydrolysis. Hydrolysis is simply breaking molecules apart by reacting with water. For the hydrolysis, we're going to need 10% of H2SO4 or sulfuric acid. Then it was being conducted by placing a pinch of yeast RNA in a test tube. Add 10 ml of 10% sulfuric acid. Heat the mixture boiling for 2 minutes. Divide the contents of the test tube into four equal portions. So the results mixture with the hydrolysis will be going to use with the following tests to be conducted. So let us first discuss who discovered this test. Stanley Rossiter Benedict was an American chemist best known for discovering Benedict's reagent, a solution that detects certain sugar. Benedict test is most commonly used to test for the presence of glucose in the urine. Glucose found to be present in urine is an indication of diabetes mellitus. It is a disorder in which the body does not produce enough or respond normally to insulin, which causes blood sugar levels to be abnormally high. Benedict's reagent is composed of 17.3 grams CuSO4, 5H2O or copper sulfate pentahydrate, 100 grams of Na2CO3 or sodium carbonate, and 172 grams of NaC6H507 or sodium citrate. Benedict solution serves as reagent in this test. Then the procedures are: 1. Using a pipette, accurately take 5 ml of Benedict's reagent and slowly transfer it to test tube 1. 2. Take 5 ml of freshly collected urine by pipette and add it to test tube with Benedict's reagent. Test tube should be held securely with the test tube holder to heat it on the burner for 2 minutes. Then observe the changes. Different precipitates are formed depending upon the sugar concentrate in urine. So for the results, as you can see on the illustration below, every color of the results interprets something. For the blue color, concentration of reducing sugar is 0, and it means that no reducing sugar is present. Green PPT or precipitate is 0.5 to 1 gram. Traceable amount of reducing sugar is present. Yellow PPT is 1 to 1.5 gram. Small amount of reducing sugar is present. Orange to or red PPT 1.5 to 2. Moderate amount of reducing sugar is present. Brick red PPT greater than 2, large amount of reducing sugar is present. So for this experiment, the results turn into reddish color, which means that large amount of reducing sugar is present. The results interpretation for Benedict test means if it's positive results, there will be a formation of a reddish precipitate within 3 minutes. Reducing sugar is present. These sugars are glucose, fructose, and ribose. It is negative when there are no color change. Reducing sugar is absent, such as sucrose. Next, the next chemical test is the test for purine. The word purine or pure urine was coined by the German chemist Emil Fischer in 1884. This test measures the amount of uric acid in the blood, or urine. Uric acid is a normal waste product that's made when the body breaks down chemical called purines. These purines are a substance found in our own cells and also in some foods. This is a test for guanine or adenine, or also called purine bases. The reagents for this test are 10% of NaHSO4 or sodium hydrogen sulfate, 10% of AgNO3 or silver nitrate. The procedures are: 1. Dissolve a pinch of the prepared RNA in 1 ml water and to another test tube, 1 ml hydrolysate. Then add 1 ml of 2N hydrogen chloride. Mix and place in a boiling water bath for 20 minutes. Then add 1 ml of 2N NaOH or sodium hydroxide and 2 ml of acetate buffer. Mix and heat in water bath. Add 0.5 ml of 10% CuSO4 or copper sulfate and note formation of a bluish-brown precipitate. Add 10 drops of NaHSO4 or sodium hydrogen sulfate solution and mix. After a few minutes of heating, a white or tan flocculent precipitate forms. Positive result. A white precipitate indicates the presence of purines. Next, the third test is the test for pentose, also known as Bial's test. Bial's test is a chemical test for the presence of pentoses. It is named after Manfred Bial, a German physician. It is a test used to detect the presence of pentoses. The Bial's reagent is composed of 0.4 gram arsenal, 200 ml of HCl or hydrochloric acid, and 0.5 ml of ferric chloride. The procedures are: take 2 ml of Bial's reagent in test tube, then add 4 to 5 drops of test solution to this reagent, keep in water bath for 30 seconds, and look and observe. The results interpretation for this test are: if it's positive Bial's test, means there is an formation of blue color such as ribose sugar. It is negative if there were no formation of any other colors. Hexose sugar like glucose and fructose are generally gives green, red, or brown color product. The next chemical test will be discussed by Miss Alondra Velasco.
Thank you, Expo. Now we're gonna talk about the test for postpaid. The reagents for this test are ammonia water. Ammonia is used commercially as the main chemical for plastic products, explosives, cleaning agents, and a lot more. 6NHNO3 or 6-normal nitric acid. Normal means the normality, that is the concentration of nitric acid. Next is NH4 2M004 solution or diammonium molybdate or ammonium molybdate. It is an excellent chemical catalyst often used in the oil industry. The procedures to this test are: number 1, in test tube 4, add 5 drops of ammonia water, acidify with 2 drops of 6-normal nitric acid. Number 2, add 1 ml diammonium molybdate solution. And then number 3, heat the mixture in water bath for 5 minutes and then observe. The result of these tests are, we can see on our screen, there has a yellow precipitate or the solidifying of some particles in a solution because of the chemical reaction in the test tube, which means it has the presence of phosphate in this solution. Next, please. So here's the summary of what we can observe in the hydrolysis of RNA and acid. Hydrolyzed RNA means it has water or it has been rinsed with water, while the other one, which is anhydrolyzed RNA, means in in original state or without any water. So in Benedict's test, the hydrolyzed RNA is cloudy light blue-green, while the anhydrolyzed RNA can be seen as royal blue or negative. Test for purine, hydrolyzed RNA, cloudy white precipitate, while anhydrolyzed RNA, lime green, which means negative. Test for pentose, green-blue precipitate for hydrolyzed RNA, while light brown negative from anhydrolyzed RNA. Then lastly, test for phosphate, hydrolyzed RNA, yellow precipitate, while anhydrolyzed RNA, cloudy white or negative. Next, please. Next. So role is role in isolation in RNA. The 1% NaOH or simply sodium hydroxide is generally used as a solid or diluted 50% in a solution. It can react with moisture from the air and may generate heat as it dissolves. Next is acetic acid. Acetic acid formula CH3COOH. It is also called as ethanoic acid. It is the most important of the carboxylic acids, a dilute, approximately 5% by volume solution of acetic acid produced by fermentation and oxidation of natural carbohydrates is called vinegar. A salt, ester, or alcohol of acetic acid is called acetate. It has many functions, but it is mostly used as chemical reagent, fungicide, herbicide, microbiocide, pH adjuster, counter-irritant, and solvent in variety of industries, including but not limited to food, agriculture, cleaning, and cosmetics. Lastly, 95% ethanol. This is the highest concentration of ethanol you can get by distillation because 95.6% ethanol is an isotrope. What is isotrope? Isotrope means a mixture of two liquids which has a constant boiling point and composition through distillation, which means the vapor state has the same ethanol water ratio as the liquid state. It is used as solvent in the synthesis of organic chemicals and as an additive to automotive gasoline, forming a mixture known as gasohol, that is a mixture of gasoline and ethyl alcohol used as a fuel in internal combustion engines. Biologic function and other questions will be tackled by Miss Eunice Mosande.
Thank you, Miss Alondra. So, first one is DNA. DNA is the information molecule. It stores instructions for making other large molecules called proteins. DNA holds the instructions for an organism's or each cell's development and reproduction. So, katulad nga na sinabi ni Sir kahapon na ang DNA ay large molecule. It is a complex molecule that contains all the information necessary to build and maintain an organism. It encodes the information na kailangan ng cell para makapag-produce ng proteins. The next one is tRNA or also known as the transfer RNA. Delivers amino acids to the sites for protein synthesis. The tRNA molecule serves as an adapter between the genetic instructions written in nucleic acid sequences and the protein products encoded in genes. Ang RNA ay single-stranded lang while ang tRNA folds into a T-shape. Transfer RNA is responsible for bringing amino acids together during translation to form the growing peptide chain that will become a protein. The next one is mRNA or also known as the messenger RNA. Carries instructions for protein synthesis, genetic information from DNA. Plays a vital role in human biology, specifically in a process known as protein synthesis. mRNA is a single-stranded molecule that carries genetic code from DNA in a cell's nucleus to ribosomes, the cell's protein-making machinery. It acts as the messenger between DNA and protein production. mRNA is the RNA type discussed the most. mRNA serves as genetic template since DNA can't leave the nucleus. The last one is rRNA or also known as the ribosomal RNA, main component of ribosomes, and rRNA combines with special proteins para makapag-form ng ribosomes, which then read mRNA to form proteins. It essentially ribosomes contain rRNA and help build proteins. Next slide. What are the products of the hydrolysis of nucleic acids? Complete hydrolysis of RNA yields a pentose sugar, D-ribose, phosphoric acid, and nitrogen-containing heterocyclic compounds are also known as the bases, such as guanine, cytosine, uracil, and adenine. Yan po ang complete hydrolysis of RNA natin. The next one is DNA hydrolyzed by alkali? DNA is not hydrolyzed by alkali pH because it does not contain the 2-hydroxide ion for base catalyzed hydrolysis mechanism. Although at high pH, there is more concentration of negatively charged hydroxide ions, which pulls the hydrogen from DNA towards it and make it deprotonated. The higher pH, there is extensive deprotonation and this results in denaturation of double-stranded DNA. This alkali method is used in isolation of plasmid DNA from bacterial cell where this principle is applied. DNA is not hydrolyzed by alkali pH dahil wala siyang 2-hydroxide ions for base catalyzed hydrolysis mechanism. Ang higher pH ay mayroong extensive deprotonation at magre-result ito in denaturation of double-stranded DNA.
And I'm Kimberly K. Cabreros.
I'm Eunice Masende.
I am Serene Mundo.
I'm Alondra Velasco. Ay,
Ano?
mali, sorry. Kala ko
Wait
ako
lang.
na.
And I'm Michelle Santos.
And I'm Alondra Velasco.
And this is all for our presentation. Thank you for listening.
Boom! Ganda ng outro. Nice. Okay na yon?
Okay na yun.
Okay na yan.
Sige.
Cut mo na lang.
Oo, kakat ko na lang. End ko tong recording na.